Hi everyone, I'm Sharzad, and today in this module we're going to talk about environmental microbiology and the application of microbiology in environmental engineering. So you guys already know that microorganisms are basically everywhere. They're inside our body, in our guts, or on our skin surface as a part of our immune system. We use them to produce different kinds of food like beer, yogurt. You guys already know all this. So it's no surprise to see these microorganisms play an important role in environmental engineering processes as well. So two of the most important applications of microbiology and environmental engineering I think are in wastewater treatment plants and also in bioremediation to clean up soil and water and groundwater. Today, in this presentation, we are going to mostly focus on the application of microbiology in wastewater treatment plants. The actual goal in a wastewater treatment plant is the substrate removal. So to make it more efficient, we need to know how bacteria grow and reproduce. We are also interested to find out the growth rate of these microorganisms and how that would affect the substrate removal. Now let's take a look at the conditions and environments that these microorganisms can grow in. One of the simplest forms of these systems is a batch reactor. In a batch reactor, we usually don't have any inflow or outflow. So for growing microorganisms, we usually add a medium with substrates, and then we seed that with a culture of living cells. It can be pure or mixed culture, and then after seeding with the culture, basically it becomes a closed system. Nothing can goes in, nothing can comes out, and therefore the microorganisms start using the substrate, so the concentration of substrate usually decreases, and then the byproducts are being produced, and the cell numbers also increases. So everything is changing all the time. But because it's a closed system, we are able to take a good look at these changes and study them over time. And this is basically the first order reaction equation that you can see here. It can be applied to concentration of any compound in the batch reactor, but here if we want to we can use it for the concentration of cells. So the change in the concentration of cells over time equals mu x, and mu is basically the rate constant, and in this case it is this. If we start observing the number of cells in a batch reactor system, we're gonna end up having this graph, as you can see. So on y-axis we have log of numbers of bacteria, and on x-axis we have the time. The general shape of this graph would be the same for almost all the bacteria. It's just the type of the bacteria and the conditions and the environment and substrates would affect the exact numbers and the time that we have here. So the first stage that you can see here is the lag phase, which the concentration of cells are almost the same, constant during this phase, and the bacteria are adjusting to the new environment and the new substrates. So they're producing all the enzymes that they need to grow. In the second stage, you'll see this rapid increase in concentration of cells, and that's called exponential or log phase. And so in this stage, bacteria has adjusted to the new environment, and they're just growing and multiplying until they get to this point, which is the stationary phase. In this phase, the number of cells being produced and the cells that are dying are almost the same. So the viable concentration is constant. And at this point we get to the maximum supportable population. And from this point on, the number of cells that are dying increases, and it's so it will be more than the number of cells being produced, so we get to the death phase. So now let's take a closer look at what is happening at each step. As I mentioned before, like in the lag phase, you can see on the top right corner, the lag phase, usually bacteria are synthesizing their necessary enzymes to degrade these substrates that they have been provided with. And how long this lag phase takes usually is a function of the history of these cells, like their age, how healthy they are, the density of the population, the substrate that is being provided, and also the type of the bacteria that we have. So on the left, these two graphs that you can see on the left, you'll see the effect of two different media on one specific bacteria, which is a species of E. coli in this case. And you see that the medium that is being amended with another compound is actually working more efficiently, and the lag phase is much shorter in that case. On the right, you'll see another graph. So if you provide this exact same conditions to two different organisms, again, the lag phase would be different because that depends on the metabolism and the enzymes that these different cells have. So that is also another factor. The next step is the log phase or exponential phase. We know that prokaryotic cells multiply and grow and reproduce with binary fission, meaning that they grow in size until they get to a specific size, and then their DNA starts replicating. When the replication is complete, the cell elongates and the cell membranes form, and the cells basically get separated. So one cell becomes two, two becomes four, four becomes eight, and the number of cells are basically 2 to the power of n, and n is the number of generation. We can study this exponential phase in reactors called chemostats. These are bioreactors in which we have a continuous inflow of medium and substrate, and also a continuous outflow of waste. And if we keep this bioreactor under a steady state, we will have a constant growth rate, and therefore we can study this exponential phase better. During the exponential growth phase, if we assume there's no cell death, we can calculate the number of cells at each generation using the number of bacteria that we initially inoculated in the reactor. So it would be n sub naught multiplied by 2 to the power of n, which is the number of generation. Then we can define this parameter called g, or the generation time, which is the time that is needed for a population to double, basically time interval over the number of generations. So now if we want to linearize this, the top equation, we can take the log of these terms, and then we're going to have this graph here with this, which is linear, and we can calculate the slope of this graph, which will give us this other factor parameter called growth rate constant. Another way to approach this is using the first order model that we talked about in a batch reactor system. So the changes in cell concentration over time equals to the specific growth rate multiplied by the cell concentration. And if we rearrange these parameters, we easily get here. And as you know, x over x naught is 2 because we are calculating the doubling time. and tau d is basically the doubling time. So doubling time and generation time are the same. It mostly depends on what parameters you have. Now we want to take a look at a couple of examples to see how to use these equations that we just talked about. So the first example is if a bacterial cell count increases from 10 to the 3 to 10 to the 9 cells in 10 hours, find the generation time or the doubling time. So the givens are the initial and the final cell numbers and the time. So you can easily use the equations that we just talked about and find G here, which is 0.5 hours. This is the second example, so we're going to find X for a single cell after 12 hours with a doubling time of 0.5 hour. We can calculate the mu and then calculate the cell number based on the equations that we had before. But, well, obviously for a wastewater treatment plant, it's very hard to use like just cell numbers. We use biomass, and the biomass is usually measured using the total suspended solids or volatile suspended solids in the wastewater. The next phase after exponential phase is actually stationary phase, but during the stationary phase, as we mentioned before, the cells being reproduced are actually equal to the cells that are dying, so the concentration is basically the same. But in the next phase, or the death phase, the death rate of the microbial population is higher than their growth rate, and that causes the decrease in the concentration of viable cells. Cell death can also happen by lysis, which is basically the physical destruction or decomposition of cell. At the same way that we calculated and defined mu, the specific growth rate, we can also define B, which is called the first order decay coefficient, and it is a negative because of the loss that we have in their cell population. And we use these the decay coefficient mostly in the aerobic sludge digestion that happens in wastewater treatment plants. Now we want to focus more on substrate utilization, which is the main goal in a wastewater treatment plant. So dx over dt, which was the growth rate, is not only a function of biomass, but it is also a function of the substrate concentration. So in a batch culture that has only a single limiting substrate, which is going to end at some point, we can find a relationship between the biomass concentration and the substrate disappearance. And we can also find a relationship between the growth rate and the substrate disappearance, which is what we're going to talk about in the next few slides. So this is the first order reaction that we had from before in a batch system. And we can rearrange the equation to calculate mu. But remember that we need continuous growth in the activated sludge for our wastewater treatment plant. And therefore we are looking for a relationship between the growth rate and the substrate removal. So this guy, Monod, was able to empirically observe the relationship between a specific growth rate and substrate concentration. So he, this is the graph that he found through his studies. So as you can see, at some point in the graph we reach a plateau, meaning that no matter how much substrate you add to the system, the specific growth rate is not going to get higher. So that is the maximum specific growth rate. He was also able to define this parameter called Ks, or saturation constant, which is defined as the substrate concentration when growth rate is at its half maximum, like this. So with this graph, he was able to find this equation which can relate the a specific. Let's take a closer look at this monods graph. So based on the substrate concentration, we can divide this graph into three different sections. The first section, we can assume that it is a linear graph in the first section, and in this case the substrate concentration is significantly lower than the Ks value, or the saturation constant, and this was Monod's equation, right? And here we can assume that because S is much lower than the K value, we can assume it is almost zero, and our equation will turn into this first order reaction. In the midsection, our Ks value and the substrate concentration are comparable, so we need to use the kinetics and this equation to find the dx over dt. In the last section, the substrate concentration is significantly higher than the Ks value, so we can assume that this is 0 and this term becomes 1. And you can see that we have reached the plateau here, so mu equals mu max, and this whole thing becomes a zero order reaction and becomes much simpler to use. We also talked about decay of microbial mass in the previous slides. That was a first order reaction, and that was negative Bx. So this term can be added to monod model to account for the microbial mass decay in our system. As mentioned before a few times, the main goal of biological wastewater treatment plant is not biomass production, but it is substrate removal. So when we are writing all these equations, we have to keep that in mind and try to use terms that are indicating the substrate removal instead of biomass concentration. So under exponential growth phase, the cell growth or changes in cell concentration is proportional to the mass of the substrate utilized. So we can use this ratio of changes in cell concentration over substrate utilization, which is called Y, or yield coefficient, in our equations. So now we want to add the true yield coefficient, which accounts for substrate utilization, to our previous equations. So if you remember from the previous slides, we can write dx equals to y multiplied by ds. So in the first equation, you can see that if we rearrange that, we'll get to dx over dt actually equals to negative y ds over dt. And the negative sign is because the substrate utilization is a loss. So we also have the monod equation, and if we rearrange the first equation that you saw here, then we can get to negative ds over dt equals to mu x over y. And then mu over y is actually defined as a specific substrate utilization rate, or Q. So we get to the third equation here. And the unit for Q is actually mass of the substrate over the mass of cells times T. Now we want to put together all the equations and information that we learned so far, and mostly put together everything in terms of substrate utilization. So in the first equation, you'll see, you've seen it so many times, this is basically just the first order reaction for the batch reactor that we had before. And the second equation, we're mostly going to talk about this RS, which is defined as substrate utilization rate. So this is basically the same as ds over dt, right? Because this is the changes in concentration of the substrate concentration over time, which is the substrate utilization rate. And this equation here is basically the same as what we had in the previous slide. So ds over dt equals negative mu x over y. And we also have the monod model, monod equation. So if we combine these two, we'll get to this equation here, which basically defines the substrate utilization rate based on the cell concentration, substrate concentration, yield coefficient. So for each system, we first have to measure the mu max and saturation constant, and once we measure these two, these are going to be constant the whole time, and then the variables are going to be the cell concentration. So to connect this presentation with our next presentation, that is more about lab tools, I just want to mention that as we saw, the microbiology is actually a very useful tool to monitor the processes in activated sludge in a wastewater treatment plant, and to figure out if we need to do anything to prevent poor effluent quality or not. In the next presentation, we're going to mostly talk about different lab tools, the actual tools that environmental engineers have to monitor processes that are going on in a wastewater treatment plant or a bioremediation project, and find out if they need to do anything and help the processes be more efficient. Some of the microorganisms that we usually see in the activated sludge are bacteria. Usually these are the most abundant types that a type of microorganisms that we see, such as filamentous bacteria. Filamentous bacteria is very important to the activated sludge because it forms it helps with forming these flocks, which would help reducing the organic carbon from the wastewater, and also later with settling, because it is heavier, it will settle down and helps with separation of solid material from the clear water. Protozoa are these single cell eukaryotic cells, such as amoeba or ciliate, which usually feed on bacteria, and their count kind of shows the bacterial count as well. We have rotifiers, which are these multicellular animals, basically, which are associated with older age sludge and in high turbidity. Then we have worms. Worms also are these animals that are able to degrade more complex carbon material that the other microorganisms are not able to degrade. So what do you think it is more important when we are observing these microorganisms? So in a wastewater treatment plant, they regularly take samples from the activated sludge and check it under the microscope. They have these tables with different factors, so they keep record of the size and nature of the flocks, the microorganisms, and the type of them, and like the count of them, and like the filaments they see. So these are some of the factors that help them figure out whether or not they have a healthy, efficient sludge, and whether they need to do anything or change any conditions for their activated sludge to help it be more efficient and to prevent poor effluent quality. So in activated sludge, we can define a sludge age as cell mass over cell production rate, and we have this specific growth rate as well. So it is basically reciprocal of the mu, as you can see. In this graph, also you'll see that the relative number of microorganisms through time in an activated sludge, and you can see that different microorganisms are dominant at different stages of activated sludge age. For example, as I mentioned before, rotifiers, you can see they have this peak towards the end of activated sludge age, and bacteria, for example, are more dominant at the beginning or to the middle of the activated sludge age. So this is one of the parameters that wastewater treatment plants can use to evaluate the efficiency and activity of the activated sludge. And these are some links to different YouTube videos that I strongly suggest that you watch them. Two of them mostly show like the different microorganisms from the activated sludge samples under the microscope, but one goes through more details of how these um, samples and like using microscopy and testing these samples can affect and help wastewater treatment plants in decision making.